As you can see, well, welcome to Neurotino. As you can see, Michel is presenting today because you currently can see his entire life unfolding in front of you on the screen
I'm showing the link to the papers.
and everything else.
Oh,
For those of you who don't know, Michel is a director of research at the University of Bordeaux CNRS, leading the neuroimaging center there. And today he's going to talk about, very exciting, the wireless connectome. So, Mish, whenever you're ready,
so
thanks for
I shared
listening.
I shared two links. I'm going to talk about three papers. There is a main paper, but this has ramification to two others that came out last year. Um, we're going to talk about the C. elegans. For those of you who don't know the C. elegans, it's a worm. Very exciting there. It's a worm that actually only have 300 neurons. So that's a manageable amount of neuron to understand a nervous system. And in the 80s, they managed to map exactly the connectome of this worm. So how those 300 neurons are connected to each others. And that create 23,000 of connections between those neurons. So you see how when you start looking at the connection within a system, things become like increasingly more complicated. And they take advantage of optogenetics, which is a method to stimulate a single cell using like a genetically modified sample, like in that case, genetically modified C. elegans, to stimulate single neurons and record all the other neurons of the C. elegans. And so by doing so, this was like, what a wonderful way to have a perfect control of a neuroanatomical system because we know all the neurons, we know all the connections, and now if we stimulate one neuron and measure all the other neurons, we should be able to figure out the link between neuron connections and activity. The hypothesis was that, like, because there is a full knowledge of this connectome, there should not be any surprise, and neurons that are connected should activate where downstream neurons are stimulated. Um, but unfortunately, that's not what they found. So let me show you the figures. They all see my screen.
Yeah, Mish, do you mind closing the box on the top so we see more of your screen? There's a X under download the PDF, there's an X, so you can close that top. Under,
Do
yeah, to hide.
yeah,
There you go. Thank you.
right. But that brought me into a different place. Thank you. This is a long paper. There we go. Not everybody is dizzy now. Okay, so this is a system, and they show you a little bit how optogenetic is working. And this is a C. elegans nervous system where every single dot that you can see of different colors is a different neuron. I think I can zoom it in right here. You can see it there. Um, so this is a raw measurement where they're gonna stimulate each of the neurons over here. It's 300 neurons. I think this is just a subsample because I cannot count 300 there. And then here you have the response of those neurons. And you can see sometimes you stimulate a neuron and you have a response in other, sometimes you do not. They do some modelization and all that kind of stuff. They create, as you've seen in functional connectivity, those very large diagrams of connectome of the C. elegans with like graph theory and all that kind of stuff. But the main interesting fact of the paper is when you compare neurons that were supposed to be activated, that you can see here in the panel B, with neurons that were not supposed to be activated because they're not connected, you see that when neurons are connected, they're more likely to show an activation. than when they're not connected. But if you look at the standard deviation, there are many cases when neurons that are connected do not show an activation. And there are many cases when neurons that are not connected do show an activation, so a wireless activation. So we kind of wondered what is going on. So they took advantage again of the model of the C. elegans that you can modify genetically to study this kind of communication in C. elegans that have a genetical defect that prevents them to have vesicles coming out of neurons, so no neurotransmission. And we found that those C. elegans have a very impaired nervous system, but some, I don't know which figure it was, some connections still remains. I mean, some functional connections still remains even if they don't have neurotransmission. So the hypothesis and what they start investigating is that actually the connection that remains are related to a different mode of communication, which is based on neuropeptides. Neuropeptides are a bit like neurotransmitter. They're just bigger. They're different. They're different. The different products. They're just bigger, and they escape the neuron not in the synaptic cleft. They escape the neurons like outside of the body, and then they diffuse around. And some other neurons have receptors for those neuropeptides, and some others don't. So that's why, in a follow-up paper from a competitive group, they've been mapping in the C. elegans the connectome of the neuropeptide connectomes of the C. elegans. And so now you have a model of the relationship between the neuropeptide communication in the C. elegans brain and the different neurons of the brain. This model, the original paper, is suggested to drive more phenomenon that we can observe of resting activity, which may be related to what we can see with functional connectivity at rest. That's why we only see it at low fluctuations. So at this point, you might be like, this is great, and it's probably only occurring in worms or C. elegans, and I don't see how this can be linked to the human brain. So this other paper, which I'm trying to reach to get the buffer here. Which one was it? This other paper, I've been investigating the neuropeptides network in mice and been showing that it also exists in mice. So mammals also have a neuropeptide networks of communication in their brain. So wireless communication without connections. You spend your life studying white matter connection in the brain and trying to model the brain based on white matter connection. It feels like a terrible news. Like we've been missing like an entire part of how those neurons could communicate together. But before you decide to throw away your career and burn all your papers, you need to know that this mode of communication is based not on electrical signals like the neurons, it's based on the diffusion of those neuropeptides inside the CSF of the brain. So it is extremely slow. You're not going to see any fast interhemispheric interaction based on the neuropeptide networks. It's just local or really slow diffusion inside the brain. So it might very well be possible that this system of communication is an ancient way neurons were communicating together along the evolutionary trees have been progressively replaced by faster electric communication in larger organisms with a bigger brain, such as humans. That's it.
That was a quick ending. Chris already has his hand up. Go for it.
Okay, so I just Googled it. I went on Google Scholar,
Mm.
and it seems that there is something with neuropeptides with the guts, because there is a gut brain
Mm.
communication. So I'm wondering whether the guts could send neuropeptides to the brain, and with some of the neurons that have the receptors from the neuropeptides, you could have like a general state regulation from the guts, because there's been a lot of studies about the relationship between the guts and the brain, how it can, like, there is a feedback loop between the two. So maybe there
Right.
is also
Yeah,
that. No?
I hear you. So like the neuropeptides are bigger than neurotransmitter, and you got the blood-brain barrier that usually filter big stuff. Question is a neuropeptide small enough to get through, or does it not? Okay, is it too big and does it get stuck? I don't
Well,
know.
the first paper I see, neuropeptids. It literally says neuropeptids in the microbiota gut-brain axis.
Okay, but does it like get through the blood-brain
Um,
barrier?
I mean, I would have
Just a
to
question
read
for
to
Google.
read that, but
Yeah, I can do it, you know. Yeah, I mean, I guess I can. All right. Blood-brain barrier neuropeptides.
That's online research live.
yep.
Oh. Do
My
neuropeptides
question
cause
would
the
be,
body?
do the neuropeptides have to cross the blood-brain barrier, or the signals that result from their interaction?
Sorry, I didn't listen to you. I was reading the. What was your question?
Multitasking. My question was if it is the neuropeptides that actually need to cross the blood-brain barrier, or if it is the signals as a result of their interaction that need to get transmitted across.
I don't know. I don't work on the gut. I guess like in the context of the present paper, the activity of neurons is modulated, or a neuron can be activated without being connected to other neurons just because the neuropeptides fixate on its receptors.
But you need a
So
reason to keep this mechanism throughout evolution, right? It needs to
yeah,
have a purpose.
well, sometimes you know, for example, enkephalin is one of the neuropeptides. It's like it'll allow you to modulate the effect of pain. So I guess,
It's pretty useful.
you still need it. Um, think, and this is not my field of expertise, but you get some enkephalin running through your body when you do a long effort, such as running for a long time. But yeah, that might be it. And again, like nature doesn't throw away what is useless. Um, Not that fast, at least. If you look at the anatomy of the eye,
Yeah,
it
but
was
there is a massive. I mean, has it been showed these kind of receptors? Are they actually in the human brain? Because yeah, it's the C. elegans, right?
yes, yes, in mammals, they exist in the human brain as
Yeah,
well.
so that means it's like on a super wide range of I don't know what's the term, but then like different species on the evolution tree. So if it
Yeah,
was
it's
useless, it
everywhere.
would have been dropped by some of the
I don't
species, right?
know. I don't know. It's still like some old stuff, old useless stuff that well, that we think being useless that exist
Yeah, but
in
not shared
biology.
between a worm and mammals.
I don't know. I cannot categorically say that's because it's not used anymore, it will completely disappear in a species.
Yeah.
especially when
Well,
all the
that was
mechanisms
just
are
my
built
guess.
on top of that. I don't know. It's a good question. I mean, like in theory, we'll think it should be this way, but sometimes, all stuff remains and
Yeah.
probably has an important effect for some stuff, but that remain to be explored.
You also mentioned that the neuropeptides only facilitate slow interaction and only local communication. And I was wondering what sense you would make from it, as in, why do we need this additional system for slow and local interaction?
So local, it depends how much time you give to it. If you infuse the brain in a neuropeptide, the entire system will be infused with it. That can potentially modify your overall brain state.
Okay,
That
so
can have an impact
it
on.
is like classical diffusion. The more time you give it, the further it
That's the
will.
principle. Yeah, the
Yeah.
principle of diffusion is
Okay.
like that. Yeah. But I don't know. I'm just in frame that might have an interesting relationship with psychiatric disorders. If you can modify an entire system, inhibit or exacerbate the activity of some networks in the brain based on this slow diffusion process, not only the functioning of your brain is going to be altered or changed or different, but before you actually get back to a normal functioning of the brain or different functioning of the brain, it's going to take a long time to wash out all these neuropeptides or peptides, as we say. But
What's the time scale that we're talking about? Do you know? Like, how long are these effects? Are they longer lasting or?
it is not indicated in the papers, so I don't know. I just know that the process of diffusion is no faster than the diffusion of water molecules, so it is extremely slow. We're talking like a few microns per seconds.
And would you think
And
that
it's not guided. It's like,
free diffusion?
Really.
Would you think that this is then a redundant system that we see, a leftover evolutionary speaking, or is this a actual system in its own right that developed in parallel?
To put an hypothesis, I'll say them at the beginning of peptide, and then a long evolution because things become like, more challenging in time-wise and in terms of distance to cover. We started having like electrical and neurochemical synapses, and that was more efficient, and that progressively started by adding up to this communication and then progressively like taking over this entire system of communication in the brain. But maybe some very primitive neural communication or important things are encoded into this neuropeptide system, and that's why it's still there, like Chris has been mentioning. It must be useful for something.
Eduardo has a question as well.
Yeah, I was wondering maybe if we could consider a similar system in mammals, like the hypothalamic adrenal axis as a sort of diffusion system for these neuropeptides that could, well, there's the bloodstream involved, so it's not properly diffusion. But maybe there could be something that is still useful from an evolutionary point of view, and also an example of these diffusion systems.
Possibly, yeah. Level of anxiety, post-traumatic stress disorder, all these things might be related to dysregulated or abundant system. Or maybe it's a way to adapt to the environment and the needs of the environment. I mean, like there's tons of things to do. Neuropeptides have been discovered in the 80s. It's not like something new, but before we thought like it was a random soup where the brain were like, you know, marinating, and that could influence the brain behave, but they didn't think there was like such a fine and precise connectome of things, and that it would actually stop the communication between some neurons or activate some of those neurons.
I mean, one of the points I wanted to raise is also that a faster communication is, I mean, it's good, but it's not necessarily the best for certain types of brain behavior systems.
Right.
I mean, you might want to have some slow paced
That's
responses.
But here it's really slow. I mean, if you had to go from one hemisphere to the other, it's gonna take days, weeks. You know, it's super slow.
Well, that's not the only way of information transformation if it takes days or weeks to cross the hemispheres. Lilith has a question as well.
Thank you for these papers. They're interesting. I'm trying to understand, is it directed or random diffusion at all? Like, is there any
Oh. So the diffusion is random, but some of the neuron have the receptor and some of them don't.
So it might be directed diffusion.
No, it's still random diffusion.
Yeah.
But
And is it possible?
some receptors are on some neurons, some others. But the diffusion is random.
So can we measure it in humans somehow, or it's possible only at worm level to understand where does this diffusion go in the end?
I mean, you know, considering with some ethic consideration, we could like inject neuropeptides to people and see what just happened. That's one thing. Don't abuse it.
Okay.
But that might be interesting. Or you could use radioactive neuropeptides and see where they go in the brain. But again, you know, it's not easy. I'm not even sure there are some clear radioligands to those neuropeptides existing. But yeah, no, it's a very good question. I mean, like it made it in nature for the C. elegans, and it went fast up to the mice. Like there's tons of work to do. As a matter of fact, I wrote to Nicola Paloma Gallagher to ask her whether she looked at those receptors as well, which she said no, because she's focused on, you know, neurotransmitters. But yeah, it was exciting to see exactly what kind of network we're gonna extract. You know, is there like a limbic neuropeptides network? I don't know.
Also, the thing that Chris was talking about, gut connection, also might be interesting to look into in connection with neurodegenerative disorders, because it's coming a lot of information about that. For example, Parkinson's disease might be somehow triggered by what we eat and what we consume, and maybe this slowly, because to degenerate Parkinson it takes a lot of years, right? So maybe can be connected. I don't know.
Good question. I mean, like 6 months ago, people
No, it is.
there is tons of things to do. You can apply it at any source and any question. It is always super interesting, and there is probably some very important things to find for our understanding of the brain and for the health of people in general. So
Yeah.
that's why I wanted to present it back in 2023.
No, yeah, it is super interesting. Thank you.
Related to that, and maybe for one of the next presenters, I just shared in our Twitter group where I bounce in papers every now and then. I just shared a paper on the inflammation, somas, and neurological disorders, the mechanisms and the review. So that might actually nicely link to the point you just made. So if anyone wants to take that paper in the coming weeks, that is up for grabs. Thank you. Now, the other thing that I wanted to ask, and you already touched upon it, but I was wondering if you could straighten it out a bit, is the translatability to humans. So it's great that it works in C. elegans and mice, but if it's difficult to map in the human brain, what's the impact that we can take from it?
So this is a discovery on the functioning of the nervous system that can only be made to this level of precision. I mean, recording all the neurons and all the connections. We're not going to do that in humans anytime soon. Sorry to break it to those of you that so do it. But it's a complex system. But that allow you to have a global understanding and discovering new mechanisms that you could not explore before. Now, it's been published in Nature in the C. elegans. I mean, everything remains to be done to translate that to humans. What the takeout of this add up a layer of understanding to what you can read or results you can find in the future in your research. You can always keep in mind there is this other system that is functioning in the brain and can actually impact my patients or my healthy control in the way they will behave during the task, or they will recover, or the disease will evolve. So this is a global fundamental understanding of how a nervous system works that open up new hypotheses for you to explore in your future research or new interpretation of your results.
That is a grant-ready sentence right there. Right, that's all the time we have today. Thank you so much. Very exciting paper meet. And I look forward to seeing you all next Monday. Have an exciting week in science. Bye.